Eukaryotic DNA replication requires two different DNA polymerase enzymes, namely DNA polymerase alpha and DNA polymerase delta. DNA polymerase delta synthesizes the DNA on the leading strand continuous DNA synthesis, whereas DNA polymerase alpha synthesizes the DNA on the lagging strand discontinuous DNA synthesis. Besides these two enzymes, 6 more factors are involved in eukaryotic DNA replication. 1. T antigen. 2. Replication factor A or ofRF-A, also called RPA or eukaryotic SSB. 3. Topoisomerase I. 4. Topoisomerase II. 5. Proliferating cell nuclear antigen PCNA, also called cyclin, and 6. Replication factor ofRF-C. The process of eukaryotic DNA replication involves the following steps. 1. Before the onset of DNA synthesis, there is a presynthetic stage of 8 to 10 minutes duration for the formation of unwound DNA complex. This step needs only 3 purified proteins, namely T antigen, T-ago tumor antigen, RF-A and topoisomerase I and II. 2. The T antigen, using its DNA binding domain, forms a multi-subunit complex with site I and site II in the presence of ATP and caused local unwinding. 3. More extensive duplex unwinding occurs due to association ofRF-A and a topoisomerase with the help of DNA helicase component ofTago Topoisomerases help in unwinding of DNA by altering topology of DNA at the replication fork. 4. RF-A or SSB proteins bind to unwound single-stranded DNA. 5. The primer RNA synthesis is performed by primase, which is tightly associated with DNA polymerase alpha. 6. DNA polymerase alpha helps in synthesis of an okazaki fragment in 5:3 direction. 7. Replication factor C, or RF-C, and PCNA, cyclin, help in switching of DNA polymerases so that pol alpha is replaced by pol delta, which then continuously synthesized DNA on the leading strand. 8. Another okazaki fragment is then synthesized from the replication fork on the lagging strand by pol alpha primase complex, and this step is repeated again and again, till the entire DNA molecule is covered. 9. The RNA primers are removed and the gaps are filled as in prokaryotic DNA replication. Recently, role of DNA polymerase E in DNA replication has been stressed upon, so that 3 DNA polymerases, alpha, beta and epsilon, are now known to be involved in eukaryotic DNA replication. A. Sugino and co-workers have proposed that DNA polymerase might function at both the leading and lagging strands, since polymerase has a primase activity, whereas polymerase E and polymerase delta are involved in elongation of the leading and lagging strands respectively.